Start with this. Okay, so modeling calcium dynamics in neuron cells. So this is the summary of my talk. I'll start with the neuroinformatics, then some basic concepts of neuroscience, then introduction to chemical kinetics, then modeling calcium dynamics, diffusion, buffering, membrane fluxes. So what is neuroinformatics? So it is a combination of neuroscience and informatics research to develop and apply advanced tools and approaches which we need for understanding the structure and function of the brain. So you have the data on neuroscience. What is informatics here? Informatics is a combination of mathematics, computer science, IT, statistics, and so many things, data, uh, science, etc. So when we talk of the neuroscience, the data becomes very large because, and when the data is very large, we always look for informatics. That's why we have today bioinformatics. When we have huge data sets, then to analyze the data set, we use informatics approaches. So again, same is the case with the neuroinformatics again. So these are the three pillars of neuroinformatics, and you can see the interaction among these three here. So we have the neuroscience data and databases. Lot of experimental work is going on in the field of neuroscience, and lot of experimental data is being generated and goes on the various websites. Today, NCBI, then there are neuroscience databases also. There are societies for neuroscience, they hold it, and then there are many clinical sites, or many sites of the universities which have these data and databases. So, uh, what to do with this data then? Now, this data consists of gold mine of information and knowledge, and therefore we need certain approaches and tools for analysis. So, objective is that when we are already having so much data, can we use some mathematical techniques or software tools to perform the analysis of this data and generate new knowledge without performing biological experiments? Many things can be deduced from here. Second thing, when we are doing this thing, we can do large number of theoretical experiments on this data rather than real-time experiment. Real-time experiment requires a lot of money, energy, and time, and computational techniques are very economical and fast, and therefore we look for computational techniques. So we analyze this data, use this data. So we develop computational models of neuroscience and use this data, perform the analysis, and then we can have the simulation. So these computational models which we develop are mathematical. So they are based on general computation principles, general mathematical principles, and these are used for simulation. You simulate on the computer. So we are able to write the programs for them, and then they take the shape of software tools, and therefore we are able to simulate them using these software tools, using these models, writing their codes. We simulate them and generate the new information knowledge which can be generated without performing the real-time experiments. And then again there is a feedback from these software tools. A new data is generated. So you are generating new information by simulation, and that information again becomes data, goes into the neuroscience databases and it becomes data. So many times you will find in databases that the source of the data is given. It tells us that it has been generated by simulation or it has been generated by experimental research or theoretical research. That is also mentioned there. Uh, so we know that all the pros and cons of theoretical and experimental works, and accordingly we are able to realize that what is the authenticity of that data which is generated uh, either by experiment or by the theoretical analysis. So this process keeps on going. Then there is interaction. Many times we do some computations here without using any software tool, and that again generates new values which go to the databases. So this also we can do. We keep on doing. So it is a two-way traffic among them. Okay. Now software tools. If there is any problem with these software tools, again this is a feedback that we need more modifications in the model so that we have a better tool. So all these things are going on, and we keep on doing this around the year. So now first we talk of some basic concepts. So brain consists of neuron cells, you all know, and neuron cells they are responsible for intracellular and intercellular signaling. All the signaling which takes place in our body, from the brain to the organs of the body, it is through the nervous system, and all the information flows through the neurons. So neurons are responsible for this signaling. So the signaling goes from cell to cell, and signaling also takes place inside the cell, because inside the cell also many biological processes are taking place, and they also depend on signals. So the signals are of two types. One is electrical signal, and the second is biochemical signal, or you can call it chemical signal. So we know that chemical ions, they have the potential to be converted into electrical uh, potential. Okay, so the charge is there on the ions, negative charge, positive charge. For example, calcium has a positive charge. Calcium ion has positive charge. So flow of the calcium will generate positive charge, and that way electrical signaling can take place. So, so the areas where the electrical signals cannot pass through, the signal is converted into chemical signal, and the chemical ions flow through that region, and then again they uh, get connected with the region where the electrical potential can flow, and they uh, share their charge. Ions positively charged will give the positive charge to that membrane, from where the electrical potential can flow further. So action potential is large, and this is rapidly reversible fluctuation in the membrane potential. Okay, so it keeps on moving from positive charge to negative charge. If the positive charge moves away, so the positive charge has reduced, and it is moving towards the negative. So potential in this way keeps on fluctuating. So you can have 5 volts to minus 5 volts. Okay, so if the ions move away, they have carried away the charge with them. So that you subtract the charge from there. Then there will be ions which will be negatively charged. So they will make this potential negative. So in this way, the potential keeps on fluctuating. And in this case, at the end of the axon, there are many nerve endings. So we'll see here, this is the axon part, and this is the neuron cell. And this part of the neuron cell is called soma. Soma is the body of the neuron cell. This part whole is soma. So soma is the body of this neuron, and within this neuron, this called, inside the cell, it is called cytoplasm, and it has many organs in the neuron. For example, nucleus is there in the neuron cell, plasma membrane will be there, Golgi bodies will be there, mitochondria is present there, and endoplasmic reticulum is present here, and so many things, so many organs are there, and they have their own functions, they have their own roles. So calcium this membrane, this plasma membrane, which is covering the whole body, this is impermeable to many ions. For example, free calcium ion. Free calcium ion cannot pass through this membrane inside the cell or outside the cell. Now, endoplasmic reticulum is a storehouse of calcium, free calcium. Then Golgi apparatus also works like a store of free calcium. Mitochondria also is a kind of store of free calcium. Then free calcium comes inside the cell from outside. So I'll talk of that, how it comes, how these organs work. So first we take the major parts of the cell. So this is called soma. This is called soma, and inside this soma is the this portion, whole portion is called soma. Inside this portion, this is called the cytoplasm, and in this cytoplasm there are nucleus and these organs like ER, endoplasmic reticulum, uh, and then mitochondria, Golgi bodies, and so on. And these are the organs of the cell, and they have their own functions. Okay. Now there is a extension here from the soma. There is an extension here which is called axon. So this is the region from the signal is fired out of the cell, and here we have the synapses. This is synapses, and these tips are called dendritic spines here. These are dendritic spines, and there are synapses here. Synapses are these are called synapses basically. These are called synapses. These are sorry, these are called dendrites, and synapses are the points of connection on the dendrite. Okay, so three major components are there: soma, dendrites, and axon. Dendrites are the branches from where the signal is input, and axon is the uh, extension from where the signal is fired out, and the soma part is the region where the signal is processed, and it decides whether to fire the signal or not. Okay, so at the end of the axon there are many nerves endings. They are called synaptic terminals. These synaptic terminals, they get connected to another neuron at the dendrite. So dendrite also have these terminals here. So they connect from here. So synapse mediates the signal transmission from one neuron to another. So these are synapses are the basic points of connection of one neuron with another neuron. Neuron at the dendrite. So dendrite also have these terminals here. So they connect from here. So synapse mediates the signal transmission from one neuron to another. So these are synapses are the basic points of connection of one neuron with another neuron. Okay, so I've already explained these three parts are there: cell body, that is called soma. Second is dendrite, from where the input comes in, and then third is the axon, from the where the signal is fired. And there are particular type of neuron which stimulate the muscle tissue. They are called motor neurons. Dendrite will receive the impulses and conduct them towards the body. That is, input comes from the dendrites. I'll show you that, discuss this again later on in one of the slides in detail. So you have the connection in this way, axon over the synaptic. So there's the postsynaptic neuron on this side and presynaptic on this side. This is the picture. There's the presynaptic neuron and there's the postsynaptic neuron here. These are the postsynaptic. They are connected with the synapses here. Okay. Now these are the resting potentials. The cell is at rest. What will be the potential inside the cell and what will be outside the cell? This depends upon which ion is inside the cell and which is outside. So this is 10 raised to minus 7 millivolt and 1 to 2 millivolts outside, generally at when cell is at rest for the calcium. I'll be talking on calcium today. So resting potential in resting neuron, that is, it's not conducting the impulse. So there is a difference in the electrical charge outside and inside. And these are the channels here, and here the sodium can get inside through the sodium channel. Then there's a potassium channel, so potassium can come and go out. That's what is there. And then there is a sodium calcium, sodium potassium pump, in which sodium will come out, potassium will go in, sodium will come out. So this exchange of ions which take place. So these exchanges of the ions, or this movement of the ions, that makes the charge of the plasma membrane. So that means the electrical difference in the electrical charge inside and outside the cell by this chemical signaling. So electrical signaling is assisted by this chemical signaling actually. So the outside has a positive charge and inside has a negative charge generally. And we see here, uh, so there are different number of potassium ions and sodium ions on either side of the membrane. And when nerve is not conducting any impulse for each, then ATP molecule is hydrolyzed. It is actively transporting 3 molecules of sodium out of the cells and 2 molecules of potassium into the cell. So this exchange of sodium ion and potassium ion in this ratio 3:2 here, and this is what is done by the mechanism called sodium potassium pump. So in this way, the charge also keeps on changing. So exterior part is generally positive and inner part is generally negative. Okay, so here it hydrolyzes one phosphate radical and this becomes ADP here. So this is the chemical process which keeps on taking place inside the cell to change the uh, uh, potential uh, from negative to positive, or reduce the potential from positive higher level to lower level, and so on. So in this way, electrical signals are propagated by the chemical ions or chemical signaling. So this creates a concentration and electrical gradient for sodium and potassium, and this means that K plus tends to diffuse out of the cell and sodium tends to go inside the cell. So this exchange takes place here. Now membrane is permeable to more permeable to K plus, so K plus diffuses out along its concentration gradient. If the concentration of K is higher out inside and lower outside, then it will move out. So the results are the net positive charge outside and net negative charge inside the cell. Now why this charge is positive outside and negative inside? If the charge is same, then there is no activity. This makes the cell to perform the activity. That is, if there is a charge difference, then we call it cell is polarized. When the cell is polarized, it will become active. And when the cell is unpolarized, it is just inactive. So this is what is happening here. So you can see here in millivolts, minus 70 to 30 millivolts. This is the potential of the range of the potential of sodium ion inside the cell here. It moves from minus 30 to minus 70 to plus 30. So resting potential of the sodium ion in the cell, it is minus 70. So cell is at rest when the charge is minus 70 inside the cell. That is, this is the level of the sodium ion in flux. It is resting potential, and soon the sodium ion concentration increases inside the cell. The potential will go on increasing and can go maximum up to plus 30 millivolts, and again it has to come down. So membrane is more permeable to sodium and potassium, so there are sodium ions also, they diffuse into the cell down a concentration gradient, and entry of sodium disturbs the resting potential, causes the inside of the cell to become more positive than the outside. So then, as the inside of the cell has become more positive than the inside of the cell, the membrane is now depolarized. So when more ions will go inside, the charge of the inside of the cell will become positive. So this is what is the process which is carried out in the neuron cell: polarization and depolarization. So in this way, polarization, depolarization goes on continuously, and in this way the cell carries out its activity. So to activate the cell, it has to be polarized, and then from moving from polarization to depolarization and depolarization to polarization, this keeps on here. So there can be at minus 70 millivolt. So below this minus 70 millivolt, we call it as hyperpolarization, and here it is polarization. And so if resting potential, it depends on the resting potential how much it is, and on the basis of that, it keeps on changing its threshold and the potential. So you have the all or none principle, that is throughout the polarization, sodium will rush inside until the action potential reaches its peak value, that is 30 millivolts. And if the depolarization is not great enough, then action potential that action potential will not be fired, or that is no impulse will be produced. So to produce an impulse of electrical signal, you require depolarization. So this is called all or none principle. Either it will fire the signal or it will not fire the signal. All or none principle. So neurons communicate with one another by direct electrical coupling or by transmitting the neurotransmitters. So synapses are specialized structures for signal transduction. That is, I showed you the picture of synapses. So synapse is on the tip of the axon of the neuron cell, and that is a specialized structure for transferring the signal from one neuron to another neuron. So these synapses generally basically are called chemical synapses, and in biological psychiatry, uh, people try to analyze these synapses. They perform the studies of these chemical synapses. So synapses can be both excitatory and inhibitory. That is, one can initiate the process or excite the process, and second can inhibit the process. So both the types of synapses are there. So excitatory neurotransmitters are basically uh, glutamate, AMPR, NMDR. These are all proteins, and main inhibitory receptors are GABA A, GABA B receptors. These are all chemical compounds, or basically the proteins which are performing these functions. So excitatory synapses will cause excitation of postsynaptic potential current on the postsynaptic neuron. That means it will pass on the impulse to postsynaptic neurons. So which increases the probability of firing the potential. That is, potential is fired when this charge becomes greater than certain threshold. Then only signal is fired. Signal is fired means potential is fired. So inhibitory synapses will cause the inhibition of potential current on the postsynaptic neuron. So if inhibition takes place, it will reduce the probability of firing an action potential. So each receptor has its reversal potential depending on ions they are permeable to. So AMPER is permeable to sodium, potassium, calcium. NMDR is permeable to sodium, potassium, calcium. So on these are the things here. Same way GABA is permeable to chlorine, reversal potential around 75 millivolts. So these are all the data here. So now I'll give you the morphology of the synapse. This is the synapse. This is called synaptic cleft. So this is a action of the presynaptic neuron, and here it is postsynaptic neuron below this, and and this is the tip of the action, which is called synapse, from where it is connecting to another neuron. So when it is making connection here, there is a synaptic cleft where the electrical potential cannot flow. There is a gap. So when there is a gap of more than 10 nanometers, then the electrical potential cannot flow, but the chemical signals can flow. So what happens? This electrical current can come to this point, and from here, I show you again in another slide. From here, the electrical potential will activate all the calcium ions present here, because positive charge opposes the positive charge. So there is a repulsion. It causes repulsion. So calcium ions present here, they will start moving towards the other end. And these calcium ions will give their positive charge to this part, and from here the electrical signal will flow. So this is what is happening inside this. So it's a combination of chemical signaling and potential signaling. So potential can flow up to this point, and then from this point to this point there's a gap here in this point, and here the signaling is taking place by the calcium ions. So now generally there are certain uh, uh, in this plasma membrane, these are the things which are present. Various proteins are there: glycoproteins, glycolipids, cholesterol, phospholipids. So many things are present here, and some are permeable to some ions, some are not permeable to some other ions. So these are the causes why the cell membrane is permeable and or not permeable to any ion. This gives us the idea. So there is a transport which takes place across the cell membranes, and cells will acquire the molecules and ion they need for their surrounding extracellular fluid. So whatever they want, they will take from extracellular fluid which is present in the cell, and this is an unceasing traffic of molecules and ions basically. So in and out of the cell throughout its plasma membrane, glucose, sodium, calcium, they go inside the cell, they come out of the cell, but the plasma membrane is not permeable to them. This again, so in eukaryotic cells, there is also transport in and out of membrane bounded intracellular compartments, such as nucleus can also come out of the membrane and go inside the membrane. Endoplasmic reticulum, this is what? This is basically organ of the cell, which it can go outside and come inside. Mitochondria, so they can go out or come inside the plasma membrane. Now, the examples of these are proteins, messenger RNAs, calcium, ATP, all these things they move across the cell membranes, and there are certain mechanisms by which they move. Now that transport takes place, because I'm saying that the cell membrane is not permeable to all the ions. So the ions to which it is not permeable, then they also move inside the cell by different mechanisms. Now, based on the relative concentration, we know that the molecules and ions will move according to their concentration gradient. That is, we know that theory that the flow of solute or liquid or fluid, it flows from higher concentration to lower concentration, and that process is called diffusion. So it diffuses through the cell. So molecules and ion can be moved against their concentration gradient. So that means we say that the flow of ions takes place from higher concentration to lower concentration, but there can be flow from lower concentration to higher also, and that reverse transport is called the active transport. So one is the transport which is natural transport or standard transport, by which it is moving from higher concentration to lower concentration, but moving from lower to higher concentration, that is active transport, requires energy to move against the concentration, and that energy comes from ATP, adenosine triphosphate. Then there are certain lipid bilayers present inside, which are impermeable to most essential molecules here. So the lipid bilayer is permeable to water molecules, few other small uncharged molecules like oxygen, carbon dioxide, and they diffuse freely inside and outside. So diffusion of water through the membrane is one such important to the cell, and that's why we call it as osmosis. We have given the special name. So So membrane is impermeable, but still there is a movement here. So it's just a kind of facilitated diffusion in this. So lipid bilayers are not permeable to such ions. So potassium, sodium, calcium, they are called cations. They have the electric field. They migrate towards the cathode, negatively charged. So they move towards the negatively charged particles. And in this way, they are moving from higher concentration to lower concentration. Same way, chlorine, and these are negative. So they are called anions. They are migrating towards the anode. So their concentration may be more near the anode and less near the other end, but they move towards the other end. So then there are some hydrophilic molecules. Okay, so we have another diffusion called facilitated diffusion. In this, what happens? There are certain gates they open. So membrane is not permeable. So ion take place through protein assemblies or protein embedding. Okay, so membrane is not permeable to certain ions, but there are certain proteins to which it is permeable. So what happens? These ions will combine with these proteins. They will bind with these proteins, and then they can pass through the membrane. So such type of diffusion is called facilitated diffusion. So one is this, and second is the gated one. That means there are certain gates which become open when that ion is required in the cell or required outside the cell. So the gate in the membrane opens, and then that can pass through. And it can pass through in any direction, higher concentration to lower, lower to higher, whatever it is. But it is facilitated because the cell membrane is not permeable. Then you are giving a gate to open, and from that gate it is pushed out or pushed inside. So the there are certain pumps which can push inside from outside, and there are certain pumps which can push from inside to outside. So these are called gated mechanisms. So there are different types of gated mechanisms also in the cell. So you can see these are the gated channels, ligand gated channels. If there is some ligand which comes and bind with the gate, it gate opens or gate closes. So there are certain ligands which make the gate to open or gate to close when they bind there on the receptor. Then there are some mechanically gated channels in which the mechanical procedures are followed by that. Then there are certain voltage gated channels. So if there is some threshold of the voltage, gate will open or close. Then there are some light gated channels and so on. So there are different types of channels in this way. Vertically, they are sodium channels like here. They are fast, persistent. Potassium channels are voltage dependent. Then there are certain intracellular calcium dependent channels also. Potassium channels all depend on calcium also. Then there are certain fluoride channels. Then there are some cation channels. Then there are some calcium channels. So these are all different types of channels which are there through which the ions can pass, uh, and they are meant for particular like sodium or potassium, or they are specific to those ions. Now, about 99% of our body calcium is deposited in the bones of the and the teeth, and 1% is in the body fluids. And this 1% is divided in between diffusible calcium and non-diffusible calcium. Diffusible calcium is bound to the blood proteins, for example, albumin, and some amount is bound by globulins in the blood also. So there are two types of proteins, globulins and albumin in the blood, which bind this calcium. Then we need and use more calcium than any other mineral in the body. And another fact is there are 179 different uses of calcium in the body. It controls our muscle contraction, relaxation, responsible for nerve impulse transmission, transfer of information between the brain cells, and so on. Then it is controlling the osmosis, diffusion through the membranes. Then it passes the information within the cell. Then it will control the formation of enzymes, hormones, DNA formation in chromosomes, and so on. So many functions are there for this calcium ions in the cell. They use in blood clotting, urine filtration, formation, maintenance of bones and teeth, and so on. So many functions are there for the cell. So where does the cell come from? Ca, calcium comes from. How Ca is increased? It is cytosolic. It is in nanomoles, say 100 nanomoles to 1000 nanomoles. And if I take this here, when it goes beyond certain threshold, it becomes toxic. So if it remains for longer period of time, it becomes toxic. For shorter periods, the rise in the calcium concentration for shorter periods is not toxic, but there is a threshold of time after which it will become toxic, and it will lead to the cell death. So it should not remain high for a longer time, and it should fall down. So there are mechanisms for elevating the calcium concentration, and there are mechanisms for de-elevating the or reducing the calcium concentration. Now, where the calcium goes, if I am reducing the calcium concentration, how it is reduced? So there are calcium elevating mechanisms. How we can increase calcium in the cell, and there are mechanisms by which we can decrease calcium in the cell. And this increase or decrease of calcium is balanced, and that is called regulation. And it is regulated. So we want to regulate the calcium at certain concentration level. So we increase or decrease we maintain the balance between increase and decrease of calcium. That is calcium elevating mechanisms and calcium reducing mechanisms. So you have the calcium dynamics here. Agonist is there. It comes here at the receptor, and from receptor, okay, I'll go back here. It comes here at receptor. From here receptor, it releases the IP3 molecules which will come here. They will bind here. It will open the gate. This internal store ER. IP3 receptor is there. So IP3 will come here. So here there is some glycoproteins. They use this for generating PLC, phospholipase C activity, and from that IP3 is generated, and there's another called DAG is generated, and then PKC is generated. So these are the various chemical compounds generated, and they have the role in creation of IP3, and then they bind to IP3R and it opens the gate and calcium flows. I'll show you later on. There are calcium, sodium, calcium exchanges there. There is a ATPase by which it can move the calcium out. There are calcium channels by which calcium come from inside to outside to inside, and there are ATPases from by which calcium moves from inside to outside. So this mechanism we'll see just now. So there are certain neurotransmitters, sensory inputs, hormones, energy. So all these are responsible for open or close the permeable channels, and then cytoplasmic calcium signal is generated, and it is sensitive. There are certain switch proteins like calmodulin, troponin. They bring changes in these activities like contraction, secretion, channel gating, metabolic signaling enzymes, gene expression. All these functions. In these functions, they are sensitive to these compounds and their activity. So this regarding the calcium metabolism, there are ligand gated channels, sensory and other channels, voltage gated channels, and so on, by which it goes. And I've already explained this ER, Sarka pump is there by which goes outside. So I'll take up that later on again. So there are mathematical models given for these processes. And the first model was given by Hodgkin and Huxley, for which he got the Nobel Prize. And later on, we have a reaction diffusion model, and then there are other models. So I'll take up this one by one. So the question is, what is a chemical reaction at a molecular level? So computation of chemical constant, forward and backward binding rates. With the help of this, we find this forward and backward binding rate. So chemical reaction will have forward action, backward action also, reversible also. So forward, backward binding rates. On the base of that, we compute this thing. Then we have reaction diffusion equations. We analyze the role of the cell geometry. Then we can perform the noise analysis. That is, noise means there are errors in the uh, mathematical techniques. So we perform how much error is the error in those. So we do generally solve the partial differential equations and stochastic partial differential equations and try to approximate them, and then we see how much is the error in them. Now, noise potential, it is a concentration ratio for specific ion inside and outside. So today I'm not interested in this noise potential. It's basically on the electrical potential, and the voltage is calculated by this formula. Nernst has given this formula for electrical potential, which is an input to the neuron cell. So we'll not talk much on that. We are more focusing on the calcium dynamics. So uh, so I'll not go into the electrical potentials of this membrane right now, and so I skip these models, ohmic models, and I'll come to the calcium channels. So Horton and Huxley were lucky that they started the giant squid axiom. They talked of the sodium and potassium ions, and uh, they gave that model which got the Nobel Prize. Then Ca2 is critical for life and death of neurons. So uh, many functions including electrical signaling, gating, transmitter release, enzyme activation, metabolism, all depends on the calcium signaling here. So calcium currents are modeled using the GHK current, that is Horton-Huxley models GHK current equation, and it has some ohmic relationship between the voltage and current we have seen earlier there. So this is to reflect the enormous concentration gradient across the membrane, basically. Uh, this thing. Calcium currents are modeled using GHK current equation than the simple ohmic relationship between voltage and current. So this reflects how much this concentration gradient across the membrane is there, which can restrict the outward flow of calcium. Even this may be electrically favorable here. Now, Ca2 accumulation can serve rough estimate of past history of spiking in the cells. How much calcium was in the past of the cell. So Hodgkin-Huxley model was able to calculate these values, and he gave an exponential model for this as a solution for the K channel. So he modeled these channels actually, K channel and sodium channels, and he was able to give the voltage levels in the cell. Then you have the transient conductance here. Again, these are based on the model of Hodgkin-Huxley for the sodium channel, and these are the calculations. I'll not go into details of this here right now. This is the Hodgkin-Huxley model actually, and so it depends on the voltage, rate of change of voltage in the cell. That depends on various parameters. But I am focusing on calcium dynamics, so I'll not take up this again here. Just I wanted to give you a glimpse of that. So similar the reaction diffusion equations are used for studying the calcium dynamics in the neuron cells. So you can see here rate of change of calcium concentration, Ca2 plus is calcium concentration. Rate of change of calcium concentration in the neuron cell will be equal to the quantity of diffusion dCA del square Ca2 plus. This is the diffusion uh, equation, and this is a reaction term. First one is the diffusion term, reaction term, and this the fluxes. The flux can be influx or outflux. So flux is calcium moving in the inside the cell or calcium moving out of the cell. So reaction term is minus Kj plus Ca2 plus Bj plus Bj plus Kj minus Ca Bj. Here means Bj is the free buffer and Ca Bj is the bound calcium bound buffer. So we know that the free calcium in high quantity for longer periods is harmful for the cell. So cell maintains the concentration of the calcium at low levels, and the calcium level is increased when any activity is required, because calcium has the role in initiation of any activity of the cell, sustenance of any activity of the cell, and termination of any activity of the cell. So that it is the signal for that. So there are appropriate levels of concentration for initiation, sustenance, and then termination. So we need some free calcium for that, and that free calcium is raised to that level for that particular function. And when the function is over, this calcium will bind with the buffer, and it will reduce the free calcium concentration in the cell. So Ca bound buffer, calcium bound buffer. Now we have free calcium and free buffers. They will bind together here. Now this is minus Kj plus. Kj plus means association. So Kj plus is association rate, association of Ca2 plus and Bj. This will form what? Bound buffer, calcium bound buffers. And the calcium bound buffers which have been found earlier, there will be some dissociation rate. This will dissociate Ca and Bg to form free calcium and free buffer. So Kj minus is, this will be in the, actually this minus should be on the superscript here, and this slide is not showing, and plus should be in the superscript. It's not Kj plus. Kj plus, this plus is in the superscript actually, and this minus in the superscript here. So that represents that it's association, and this is dissociation. Minus means dissociation. It is separating the two here, and it is combining the two here. So what will happen when these two will combine? The concentration will become negative. Which concentration? Free calcium concentration will reduce. Okay, so that's why it is minus. So if free calcium binds with the buffer, so the free calcium concentration will be reduced by so much amount. That's why it is minus Kg. And when we dissociate the calcium from the calcium bound buffer, then this is dissociation. This Kj is minus is in the superscript here. So this will dissociate the two. So this will add free calcium concentration in this. So that's why it is positive. So this is coupled to various ordinary differential equations uh, like receptor rates. It is rates actually. IP3, diffusion, buffer, calcium inside the ER, mitochondria, and many other things. I showed you those organs there in the cell. So how they are coupled and what are their specifics, they depend on the particular model which we are using here. So we assume the uniform calcium flux across the compartment membrane. Then we have to take only one dimensional diffusion, that's the radial diffusion. The fixed law of flux, first law for the flux is JCA minus alpha DCA del C A by del T. So this here is the concentration, sorry, del C A by del R. It's R, sorry. So this is a concentration gradient here into diffusion coefficient of calcium, and there's some constant here. So any parameter diffusion constant is dCA, which will depend on the ion size and the medium. So dCA of water is 600 micromoles per square per second, and cytoplasm it will be 200 micromoles per square per second, and so on. Then the buffers, they bind with the calcium. This is a chemical reaction which is taking place. This is association. This is the correct one. The previous one was showing some mistake here, plus here. This is the right way to write. So flux is caused by this reaction, Ca plus Bg. So association is taking place, it is forming the calcium bound buffer, and dissociation takes place, it forms the free calcium free buffer. So in this Rj is like this. You can see here for association the two are combining, dissociation is breaking the two. So if these are combining, they form bound buffer, so this will be subtracted from here, and this will be added here. So there are some backward forward constants here. These are called backward forward constants basically. And Vg take is the total buffer. Total buffer is equal to buffer plus calcium bound buffer in the cell, and uh, diffusion constant of buffer is dBj. Then the dissociation constant is calculated in this manner. Then buffer capacity is based on this kappa. This is called rate of change of CAB upon with respect to Ca2 plus free calcium. And this comes out to be this formula. And beta is called the buffering factor. That is how much calcium you are buffering down. So free calcium upon total calcium. Total calcium is actually free calcium plus calcium bound with the buffer. So this will lead to basically 1 upon kappa. Then you have a rapid buffer approximation in which buffer binds very rapidly all the calcium and becomes saturated. For that, these constants are given. So we come down to the final equation, that is rate of change of calcium with respect to time in the cell depends on is equal to the diffusion of calcium in the cell minus the flux and then you can add some buffers also here, that is reaction term. So uh, these are some approximations which we do. Generally calcium concentration is very less than capital K, so kappa can be taken in this way and beta can be taken in this way. So our equation becomes takes this form: dCA into D effective del square Ca2 plus this is what from we are taking for this for rapid buffering approximation basically. So I'll come to the simplified equation for you all. So finally, for excess buffering, we generally were excess buffering is more better simplification than the rapid buffering, because in the cell buffers are able to move outside the membrane and inside the membrane, uh, inside the membrane from outside and outside from the inside also. And buffer can bind calcium and carry with them outside, and they can bind with the calcium, bring calcium from outside to inside. So buffers are basically those proteins which can work for facilitated diffusion of calcium. Actually, what I explained earlier. And this excess buffer approximation was given by Nair in 1986 and Smith in 1996. And this gives a basic simplification of our model. This is a reaction term. So it is saying that this takes place by buffering, uh, and this is the quantity, the calcium at rest and free calcium at any time. This difference is bound by the buffer and finds the bound calcium, and therefore this amount is subtracted from this term. And there are certain fluxes which could be influx or outflux. So it will contain the three terms here in this way: diffusion, reaction term, that is buffering, and third is fluxes. So this is just a del square. What is del square? 1 upon r square d by dr of r square d by dr. This the operator along the radial direction, difference operator. And these are the buffers. There are different types of buffers: slow buffer, fast buffer, endogenous buffers are there. You can see their activity level. Accordingly, you can decide whether they are fast or slow. And these are the names of those buffers: calbindin, calmodulin, and many diseases are there due to the defects in these buffering processes. So buffers are very important in this study, and there are 50 known buffers such type. There are 50 known proteins which can act as buffers. So there are EGTA buffers, BAPTA buffer, Flura, CA green. So this is slow and this is fast. Association is very slow, association is very fast. So buffering process will be fast. BAPTA are fast buffers, EGT are slow buffers in this category, exogenous. They come from outside, they are formed outside. So generally the agonist comes here at the receptor, and then glycolysis takes place. It leads to PLC at beta phospholipase C. This will generate the IP3, and then there is also PLC taking place to generate IP3. IP3 binds with this gate at the receptor called IP3R of the ER, endoplasmic reticulum. This endoplasmic reticulum is the internal store of the calcium. Once this binds here, the gate opens and this calcium will come out. I'll show you in the slides. And accordingly, we can have the equations also. Calcium within the cytosol will have these equations. Calcium in the ER will have this equation. And IP3R will be this equation. And IP3 here. IP3 receptor will have this equation, and IP3 has this equation. So, uh, this leads to a system of differential equations essentially at the temporal scale right now here. So for calcium model again, there are certain details you can have for the cytosol. This I've already explained in the previous slide. I will not go. So we'll take up the model and try to study how we can study calcium diffusion in the cell. So we have got the basic model up till now based on the biological process. So what I was explaining is that, see, there is a gap here in the synaptic cleft. And what is happening here? This electric current will come here. It will activate the ions, chemical ions, that is calcium ions. And then calcium ions will move towards the opposite surface, and here they will release their positive charge to this membrane of the opposite surface, and then the electrical current flows here. So electrical current does not flow inside here. Electrical current flows up to this point and from this point, and this bridge is provided by the calcium ions transport in the synaptic cleft. So wherever there are gaps greater than 10 nanomoles, you see here, gaps are 10 more than 10 nanomoles. So chemical ions, calcium ions are moving towards the opposite surface. And here they give the positive charge and electrical current starts flowing. So this is how the signaling takes place. Calcium goes inside, calcium from the intracellular store comes out, and it is pumped back here. Some calcium is pumped outside the cell. I'll show that just now in another slide actually. Yeah, this is there. This is the picture. You see, at rest, calcium is generally 0.1 micromoles inside the whole cell. Okay, and calcium concentration is very high outside the cell. And the cell is not permeable to free calcium ions. Now what happens? This is a calcium channel. When calcium is required here to start the activity of the cell, initiate the activity of the cell, what will happen? The calcium ions gate will open and calcium ions will start flowing in. And in this way it will raise the calcium concentration inside the cell. So when calcium concentration increases inside the cell, the positive charge inside the cell will increase because Ca is 2 plus positive ions. Then another mechanism is there, that is internal store. This ER is endoplasmic reticulum. It is the internal store of calcium. Now what will happen? There is a IP3R receptor here, and this is called a gate from where calcium can go out here when required and come inside when the activity is over. Now there is a agonist. This again another receptor. This agonist which comes here and it binds with this receptor, and when this binds with the receptor, IP3 receptor here, it will release the three IP3. It will release some IP3 molecules, and these IP3 molecules will start moving towards the IP3R receptor of the ER, and the gate opens. So one IP3 ion and one calcium ion, they come here, bind, and they open the gate. When the gate is open, the calcium concentration from internal store will come out into the cell, and it will become more than 10 micromoles. And what it should be less than equal to zero point 0.1 micromoles. So it cannot remain 10 micromoles for longer period, otherwise the cell will die. It will toxic to the cell. So one calcium ion will come here and it will block the gate. So movement of calcium ions from inside to outside will not take place, and then there are certain pumps which will start pumping the calcium ions, and it's again becomes zero point 0.1 micromole in the cell. Now we come to the mathematical formulation. So you get these equations. So you have an equation for calcium, the diffusion term, reaction term, and the source term, the calcium channel from which calcium is coming in. And this is the term for the free buffer, the diffusion of buffer in the cell, reaction term. So this equation I have already explained. Now we are going to use this equation. Okay, we are going to use it. So what we do is we perform excess buffering approximation of reaction diffusion equation, and it takes this form. This was the final form we got earlier. Now we define the initial and boundary conditions for this. So calcium concentration at time t equal to 0 is assumed to be Ca2 plus infinity, that is resting calcium. This is the value of resting calcium. This is about how much? 0.1 micromoles. We have seen in the cell. Resting calcium is 0.1 micromoles. And limit x tends to infinity, far away from the cell, the calcium concentration is assumed to be constant at 0.1 micromole, that is resting calcium. Okay, and then there is a source sigma from which the calcium is coming inside the cell. It is moving from outside to inside the cell. So limit x tends to 0. We assume the source at x equal to 0. So at x equal to 0, there is influx of the calcium through the system. So we consider the cell to be a circular cell, and we divide the cell into large number of elements. Say here it is divided into 60 elements here, and we are using the triangular ring elements here. These are the triangular ring elements. So in this way, finite element procedure is a very effective procedure to perform the numerical studies of any problem. So we are able to divide the region into so many elements, large number of elements, uh, and then for each element we can perform the study separately and then assemble to give the results for the whole region. So variation form, there is a variation form which is created by using the Euler-Lagrange's equation is there. So the differential equation is converted in this variation form here using the Euler-Lagrange's equation, and we have discretized the region into the triangular ring elements you have seen there. So for each element we evaluate this integral. So evaluating these integrals, you will get certain values. So to evaluate the integral, you need the interpolation function. So u will be represented by some interpolation function. You must be studying interpolation in your basic course in numerical methods. So these are interpolation function, and this is we substitute in place of Ca in our equation. And here any, these are the values. They come out to be like this. These are the interpolation functions, and they are called the natural coordinates also. Uh, Basically, they are the normalized coordinates actually. Uh, then this is a procedure of finite element where we extremize with respect to each nodal concentration. The integrals are extremized with respect to each nodal concentration, and you will get a system of ordinary differential equations here. Sorry, this is a it will lead to ordinary differential equations uh, where these are the values of the integrals. and then we assemble them in the matrix equation for each element. So finally we'll get this ordinary differential equation, system of ordinary differential equations. So performing the calculations, we have k for each element calculated like this. For 60 elements we have all this here, these things are arranged here. So all these calculations, we put them in, we give them notations here. This part is represented by k, this part is represented by m, and so on in the matrix form. Now these are the values of various values of the parameters which were taken from the literature, and these are used to perform the calculations here in these equations. Now you can see the results. This shows the calcium profile. It is falling down. So near the source, the calcium is maximum. And as we move away from the source, the concentration is decreasing. So the calcium concentration decreases with respect to space, and it is decreasing along x-axis as well as y-axis. You can see it is moving like this. So this is y-axis, this is x-axis, and this is the point where the source x equal to 0, the source was situated. So here it is x equal to 0 and y equal to minus 5. We are taking -5 to +5. Okay, so this is the other end. If we are taking circular, then one side is -5, another side is 5. Okay, radius will be 10. So in this way, uh, the calcium concentration is maximum at the point source, which were taken to be at x equal to -5, and then it goes on decreasing away from the source. So you can see how the diffusion of calcium is taking place along x-axis along here. It is falling down. Then with respect to time, at different spatial locations, the calcium concentration is increasing. Because of the source influx, more and more calcium is being added into the cell. And so with respect to increase in time, the calcium concentration will go on increasing. Now again, we have the effects of various parameters at time t equal to 1 millisecond. This was the behavior of the calcium distribution in the cell, and at t equal to 10 millisecond, this behavior changed. Further, there is a change in behavior. You can see from here. Further, there is a change. See from here. So this is for different time points: 50 millisecond, 1 millisecond, 10 millisecond, 50 millisecond, 100 milliseconds. Now you see how the calcium concentration is changing along the circumference, this along the circumference of the cell, and this along the diameter of the cell. The behavior is different here. Then again you can see at angle theta equal to 0, this is the curve, and at angle theta equal to 30, this is the curve. Why there is a difference here? Because the source is placed at theta equal to 0. Source is placed at theta equal to 0. At theta equal to 0, r is equal to minus, x is equal to 0 and y equal to -5. So this way, you can see there is a significant change. Now at different points, x equal to -5, x equal to -3, you see as we are moving away from the source, the concentration is decreasing here. And with respect to degrees also, concentration is decreasing. Now this is for different buffer values. When the buffer value was 50, maximum calcium concentration is 10 micromoles. And when buffer value is 100, the maximum concentration is 15 micromoles. And when it is 150, it goes beyond this thing. Sorry. For buffer value, higher buffer value, it will go on decrease. It's basically for sigma, I think. Something is wrong in this figure. Yeah, for different values of sigma. Now you see here source influx. When the source influx is 1, it is 1.5, between 2 and 1.5. When source influx is 2, it is between 3 and 4. It's just double of this. It is between 1.5 and 2, and it is between 3 and 4. So this is just double of this. So the calcium concentration is increasing in the ratio of the source influx. You can observe from these figures. Okay, now this is a calcium concentration along x and y direction for different types of buffers. Okay, first one is for BAPTA buffer. Tell it as namely BAPTA and EGTA. This is EGTA. So for the slow buffer, the fall is small, and for the fast buffer, the fall is faster. The fall is more. Yeah. Now again, I can put this equation in this form along the cylindrical coordinates here for cylindrical shaped neuron, and then I can write the variational formulation again for that. So now in this case, we try to combine the Fourier series with the finite element. So along angular direction we apply Fourier series, and along radial direction we apply finite element. So this is a combination of two techniques to solve the problem here for two-dimensional case. So these are the interpolation functions we use here, and we find the integrals and then extremize the integrals. This is the procedure of finite element method, and then we get a system of differential equations like this, ordinary differential, and we apply the Fourier series here. So using Fourier series for this function, we get the Fourier coefficients here, and you can solve these equations to find the Fourier coefficients. Okay, so all these are substituted, and we get the matrices which are containing the components as Fourier coefficients. So we draw the picture here. You can see calcium maximum is where? At pi by 2 or pi. This concentration is maximum where the source is present. So I think source is present at pi. Yeah, pi. So at pi it is maximum, and at other points it is lower. This again the similar case, and this is for different value of buffer.